joining us, and welcome to our Wednesday webinar series, which, as you know, takes place on the fourth Wednesday of every month. I'm Dr. Elisa Fuentes Montemayor. I'm a senior lecturer in applied ecology based at the University of Stirling in Scotland. And I'm a former board member of Bats Without Borders, and I am going to be your host for today's webinar. Before we start, I would just like to remind you that our webinars are free to everyone, and just to ensure that everyone can join. But we have limited funding to run these webinars, so we ask you, if you are in a position to donate towards our Zoom costs, we would be very grateful. And there are various ways to donate, and you can become a friend of Bats Without Borders, or you can give a one-off donation, and we're going to be putting the links to our donations page in the chat shortly. You can also follow us on social media and find out more about our upcoming webinars and other events via our webpage. And you can also catch up on any previous webinars you may have missed on our YouTube channel. On your screens, you can see now a QR code, which links to our Bats Without Borders WhatsApp group for anyone who would like to keep in touch with our latest news and updates. So you can just scan the code if you want to join the group. Our webinar series runs from March to November, and here are some of our upcoming webinars, so you can get the dates in your diaries if you can join us. And just before I hand over to our speaker today, I'd just like to remind you that for sound quality and internet stability, we ask you to please keep your microphones on mute and your videos off. We welcome your questions, so please write them in the chat through the talk, and I will read them out at the end, or you can at the end unmute yourself and ask your question. So it's my great pleasure to welcome Oli Aylen as today's speaker. Oli is a PhD candidate at the University of New South Wales and a visiting fellow at the University of the Sunshine Coast. His passion for bats began in New Zealand, where bats are the only native terrestrial mammals. And during his undergraduate studies, this was during his undergraduate studies at the University of Otago. He developed his bat research skills and bioacoustic expertise during his MSc project in the pristine rainforest of Brunei Darussalam in Borneo. Oli's first hands-on experience in Australia came as a volunteer with the Australasian Bat Society's Cape York Bat Blitz, where he spent several weeks trapping bats with a larger research team. And that trip confirmed his desire to work in Australia, leading him to take a position with the Australian Wildlife Conservancy. Oli is now in the final year of his PhD, investigating whether bats benefit from fence conservation reserves, and his first thesis chapter has recently been submitted for publication. And today he's going to be talking to us about Arid zone bats and fence conservation areas. So Oli, welcome, and thanks very much for joining us. And I'm just going to hand over to you now for your presentation.
Thank you very much, Elisa. I'll just put my... Oh, fantastic. So I hope that's working and you can see that very cute bat on the screen. So yeah, thanks for the introduction.
Sorry, Oli, I think we can see your presenter view rather than the full screen.
How's that?
That's in full screen now. Perfect.
Yeah, awesome. Cool. So thank you so much for coming along. Yes, today I'm just going to talk to you about my PhD so far. I've got about a year left, so it's sort of in development. First chapter's ready, but the others are still being developed. So yeah, happy for any input, questions, comments, etc. And yeah, so my broad research question is, do arid zone bats benefit from fenced conservation reserves? And that's working at two sanctuaries that I'll discuss in a second. And we've come at this with a multi-method approach to try and answer this question. But of course, with any research, you generally come up with more questions than you actually answer. Before I get started, I'd just like to acknowledge the Wankamara and Kokatha people and their ongoing connection to this beautiful country, which are the two reserves that I've been lucky enough to work on over the last 3 years. Now, many of you may be familiar with these very large fenced areas that we build, especially in the arid zone of Australia, but also in New Zealand and other places in the world. And essentially, these areas restore the parts within the fence to conditions pre-European settlement, particularly in New Zealand and Australia. So on the left is inside the fence, and on the right is on the outside. And of course, the main thing that we're doing here is we're removing the feral predators, particularly feral cats. But we're also removing the larger herbivores from the landscape. So macropods, so kangaroos in Australia are removed from these areas. And of course, rabbits are a big problem. So they're also taken out. So there's lower predation pressures. There's also there's no predation pressures from ferals. There's also lower herbivory, apart from the native animals that are then reintroduced into these areas. And of course, these areas are generally built for terrestrial mammals. They can't get in and outside the fence. This beautiful animal here is the bilby. This is an iconic animal of conservation significance in Australia. In many states, it's been extinct for almost a century, apart from within these fenced areas. And generally, the benefits of these fenced areas are pretty successful, particularly for these ground-based terrestrial mammals. They thrive in these systems, and there are a whole network now, sort of over 35 of these fenced areas across Australia. But no one's ever looked at whether these areas benefit bats. So I took on this project, and we're looking at 2 fenced areas within Australia. So it's a bit of a busy map, but essentially these white lines are the state borders. So you have Queensland up here and New South Wales here. If you're not familiar with Australia, sorry, I should have put a whole map of Australia up. It's sort of the bottom right-hand corner of New South Wales. And where you have the 3 states joined, so South Australia, Queensland, and New South Wales, in the corner there, we have wild deserts. So this was established in 2018. It's a partnership with the New South Wales government and the University of New South Wales, where I'm a PhD student. And this is 4,000 hectares. And we have 5, I think that's now actually up to 7 reintroduced mammals just this year. And then we have arid recovery. So this is out in South Australia. It is slightly larger, so over 12,000 hectares, and there are 6 reintroduced mammals that thrive in this sanctuary. As far as their ecosystem type, they are very similar. They have similar amounts of rainfall. It's about 200 mil annually. And the vegetation is similar, but wild deserts is generally a lot more treed. So you can see from these pictures, it gives you a bit of an idea. Wild deserts has much more of these mulga dunes. So the mulga is an arid specialist tree. It's across most of Australia. Whereas arid recovery is a lot more sort of shrubs and sparser tree cover. However, the main difference that we're looking at here is the age of the reserve. So wild deserts has only been established since 2018. Whereas arid recovery has been established since 1997. So that's one factor that we wanted to look into, is sort of how long the reserves have been established. Now, at these 2 sites, I had 3 main species, or 3 main focal species. There are 7 to 8 species that occur on the reserves, but these 3 are, first of all, they're the most common, but they also represent different foraging guilds. So for those of you that aren't familiar, foraging guild is what sort of space the bat occupies, and it's classified by a combination of their echolocation, but also their wing loading and aspect. So you have Camenolobus sculdii, which is an edge space foraging bat. So they're generally on the edge of the forest, obviously. We have clutter adapted species. So this is Nyctophilus jeffreyi. And both of these species are also very widespread. You pretty much find them anywhere in Australia. And then we have a more specialist species, so this open space bat, which is Ozymops petersi. So this is a little free-tail bat, and this is an arid zone specialist, so you only find them sort of in more inland Australia. And they're really interesting. They have the widest body temperature range of any known mammal. So their body temperature has been recorded at about 3 degrees up to about 45 degrees. So It's actually more than camels, and they're extremely cute bats and very, very interesting. So those are the 3 different species, but also representing different foraging guilds. So my first chapter, and this is the one that has been submitted, is looking at halo effects. So I'm sure you're all kind of familiar with halo effects, or the idea of a spillover effect. So when you restore areas, either within a fence or just if you restore a forest or an area, the benefits of doing so generally spill over into the wider environment. And this has been well documented, particularly in Australia and New Zealand, a lot of this research. There's also been some in Africa as well. But this example here that I've got, so this is a kaka on the screen. So this is a New Zealand parrot, which are at threat from possums, but also cats. And for example, in Orokonui Eco Sanctuary, which is in the south of New Zealand, they've been restoring the populations within one of these eco sanctuaries. And then we see the benefits of the kaka spreading into the wider landscape. So it's a really nice example. And there are other papers that have studied this, some in Australia by some of my supervisors as well. So we wanted to look at this and see, well, do bats exhibit this halo effect? Are there more bats inside because there's less predation pressure? There's also less herbivory. Are they utilizing these sanctuaries? And then we're seeing, or what we would expect to see, is a decline in bat activity as we move further away from the reserves. So we utilized bioacoustics, and we set up these very large bioacoustic arrays. So on the left, we have wild desert. So this is the one in New South Wales. So this was 49 detectors, and they were out for a week in summer a couple of years ago within just the North Paddock, so just in the 2,000 hectare area. We then, at Arid recovery, we had an array of 70 or just under 68 detectors. They were out for a bit longer. It was meant to be 7 nights, but we actually got hampered by rain, which is a bit of a theme throughout my Arid zone PhD. And these were out for just under 2 weeks. So, and you can see that they're arranged in increasing distances from the reserve. That is what we analyzed. And we got the data back and we ran some generalized additive models. Now, these plots are very confusing, I know that. So on the y-axis, we have predicted number of bat calls from our GAN models. On the x-axis, we have distance from fence, so 0 being the fence line, and then obviously anything under 0 is within the fence. And then on the z-axis, we have time of night, so from sunset to sunrise. And so what we're doing is we're tracking the bat activity spatially and temporally. The plots in the grid on the right is hourly activity, or sorry, bi-hourly activity, so every 2 hours. And I'm just going to point out some of the sort of major findings. So if we look in the top left, we have Nyctophilus jeffreyi, that's our clutter adapted species. And what we can see here, first of all, if we talk about these grids, we can see that in the first hour of the night, so when bats are emerging from roosts, we have these there's only 2 sites where they're first emerging from. And what's very cool is that actually correlates with our tracking data. So those are known roost sites. So we can sort of see from the bioacoustics where they're roosting, which is pretty cool. But the main pattern that we're seeing here is that we have these 2 peaks of activity around sunset and sunrise, both inside the fence and then declining away from the fence line. We have a bit of a hump in the middle there. And so that is during the night, they're foraging just outside the fence, but then they're returning later in the night. So we sort of saw this and we were like, okay, fantastic. We have a clutter adapted species which don't fly as far. There's sort of a bit of evidence here for a potential halo effect. For the other 2 species, we saw basically the complete opposite. They seem to largely be using areas outside of the fence and actually spending very little time inside the fence. We also, you'll see here, we have this clutter index in the results section here, so a significant effect. And that was just a basic on-ground habitat complexity measure. And so this makes sense for the clutter adapted species. That was the only species that had a significant interaction, well, not interaction, but a significant effect of the clutter index. So we then thought, okay, well, fantastic. At the newer reserve, we have evidence of a halo effect from our clutter species. But then when we look at a reserve that's been established for nearly 30 years, we see the opposite effect in most ways. So there's no evidence of this sort of declining activity from the fence. Our clutter bat is again actually spending more time away from the reserve. Our arid zone bat at the more established reserve seems to actually be utilizing the reserve area more so. And the edge space bat seems to be utilizing pretty much the entire landscape. There's no real trend towards either. You'll notice from the results as well that distance alone is not significant for any of our bats at either reserve. It's really that there is that strong temporal spatial interaction between the two. So really the take-home message, I guess, here is that there's actually very weak evidence of a halo effect. And our main finding really is that it's where you put the reserve. Of course, many of these roosts take a very long time to establish. So if you're already sort of encompassing areas that bats already use, then yes, there may be a net benefit from having these fenced areas. But if we're then looking at sort of long-term, these roosts take a long time to formulate. So in the short term, fence reserves probably aren't a fantastic conservation measure for the bats, unless they're already in areas that are within those fence. And we'll come back to some of the other parts of this in a second. But what you can see at arid recovery is that they are actually utilizing pretty much the entire landscape rather than any sort of trend towards the fenced area. My next chapter was looking at more of a long-term survey, so monitoring over multiple years. This was started by my supervisors before I came on as a PhD student. And the graph on the left here, the main line to look at is just the black one. And this is total annual rainfall. And what we can see here is that in 2017 was the start of a very large drought in Australia. For anyone that followed the news, we have what are called now the Black Summer bushfires in 2019. So we had, after several years of high drought, some terrible fires all across the east coast of Australia. And it was very, very dry, of course, in the arid zone. And this monitoring began in 2018. So we're really tracking the response of bats at the end of drought and then into La Nina. So I started my PhD in 2021, so after some pretty high rainfall. And so this is tracking them through that La Nina phase. And we had fewer sites, of course, because it's a long-term study. So 6 inside the reserve and 6 outside. And we tracked bat activity again using bioacoustics throughout these years. So first of all, what we can see is bat activity overall is higher inside the fenced areas, which is pretty cool. They do seem to be showing a preference for the fenced areas. Again, whether this is because of the fence or because these areas are already inside. And what we can also see is obviously how they fluctuate each year. The plot on the right is the derivative from a generalized additive model. And this essentially shows the rate of change. So for Canolobus scoldii, so our edge space bat, we actually see that there's less variation, there's less change each year within the fence, there's more stable levels of activity. Slightly so similar trend for the clutter bat, Nyctophilus jeffreyi, but then for the other species, Ozymops petersii, there isn't too much of a difference. And again, they're an open space bat. They're using a lot more of the landscape, so this is kind of expected. But overall, the activity levels are more stable inside. There's less of this large fluctuation. If we then look into more of the vegetation, so what we've used here is remote sensing data. So to try and explain, is the vegetation different inside and outside, and is this driving bat activity year on year as the vegetation fluctuates with rainfall? So there's quite a few different variables in here, and I'm still playing around with this model. One of the issues with using this vegetation data and rainfall is that it's all, of course, very collinear. So you run into quite a lot of issues with the models. But overall, what we're seeing is NPV12. So this is non-photosynthetic vegetation. Essentially, it's all of your brown vegetation, so dead wood, branches, these beautiful big dead trees, where not that sort of short-term green veg that comes up after the rain. Obviously, that very quickly then dies off and becomes the brown veg. So this brown veg, and this has been published in other papers, is a great measure of arid zone eco-health, or health of the ecosystem. Rain itself generally wasn't a great predictor. It was this vegetation alone. And then we do have some interesting interactions here The NDVI is a proxy for the green veg. It's a slightly different way of, or slightly, it's accessed from a different server. But yeah, essentially overall, this brown veg in the arid zone is a great predictor. And what we're also seeing is that it's very highly correlated with bat activity. So what I want to do now is explore this more as to sort of where all this brown veg is in relation to where the fence is. And then we can start seeing, is it because of where the fence is, or is it because it's where the vegetation is? So there's a few different things to pull apart here, but it's, yeah, great fun to be playing around with some of this remote sensing data. Now, of course, one of the biggest questions with the fenced areas is, are they being protected from feral predators? That's the major issue. That's the main reason that we build these very expensive fences. And cats, of course, really are conservation enemy number one, as I'm sure you all know. And so I had this idea. This is actually my master's supervisor, Professor Phil Bishop. Him and some colleagues did some work detecting frogs as prey in feral cats. And what they found is that they actually intentionally fed the frogs to different predators. And then they assessed whether they were detecting it when they just killed the animal and had a look in the stomach. Was there any evidence of the frogs? But then they also used DNA methods, and they used DNA methods both on the feces and the stomach contents itself. And they found that the greatest evidence came from analyzing the stomach contents with DNA analysis. So I did the same thing with cats. So we had over, I think, 62 cats that we tested. Now we know that feral cats are an issue to bats worldwide, particularly on island nations, New Zealand. There are records in Australia. They are quite scarce. But the bats in the arid zone in my area, so I've found this species, Nyctophilus jeffreyi, roosting literally 20, 30 centimeters off the ground. We know cats can hear bats. They can hear up to about 60 kilohertz. So it seems crazy to me that they wouldn't be utilizing this. You know, you find tarantulas, skinks, they literally will eat anything. So it seems crazy to me that they wouldn't be eating the cats in the arid zone. So we ran this DNA analysis. So 62 cat guts. I also trialed two different primers. So 12S is one that is often used in vertebrate prey analysis. Cytochrome B is another one I found that has been specially adapted to forensically identify bats. So I've had a go with that as well. We also have records of over 1,000 manual searches. Now, I knew the answer to this one before I started, which is kind of why I did it. They'd never found evidence of a cat predating on a bat in either of these reserves and some other areas in South Australia. But even with my DNA analysis, I've so far, and I have not finished this analysis yet, I've had a lot of issues getting my analysis done by the lab who were doing it. But so far, I have not found any evidence of cat predation on bats, which I know there's a lot of evidence mounting up against me, but it still just seems crazy to me that it's not, that they're not being predated on. But I guess for now, the bats are very happy about this, and that's great. Maybe in the arid zone, they're safe from these predators. But I think there is further investigation here. For example, as Oli said in the introduction, I used to work for Australian Wildlife Conservancy and still do a little bit here and there. I gave a presentation to some supporters of AWC while I was on a sanctuary called Scotia, which is about 400 kilometers south of one of my PhD sites. It's still arid zone. It's more semi-arid, what we call mallee country, these large eucalypts that grow from a central tuber. And I gave this presentation. One of the land managers there heard me giving this talk about how, bats, we don't seem to be finding bats in the stomachs of cats. And he was driving home one night and saw a cat up in the tree next to this nice little hollow. And so he shot the cat for me and brought it back. And we had a look in the stomach. And sure enough, we found a bat that had very recently been eaten. So I think, it is a slightly different ecosystem, but at the same time, it's very similar. It's only a few hundred kilometers away. I think we do have some evidence that this is happening. So, 62 cats is not a huge sample. I would love to explore this a bit further, whether we do try some feces as well as the cat stomachs. I think there's also factors as to when the cat was shot. This was obviously just on dusk, so as bats are emerging. So maybe that makes a difference as to how quickly the bats are identified or stay in the system. So I've just got a few more things to show you. It's not as complete. I'm in my final year, but I'm getting there slowly. So we discussed my first 3 chapters. My final chapter, this may actually become 2 chapters. I'm still working on that out, is really looking at this fine scale reserve use. So how are the bats actually utilizing the space inside the fence? And this is using several different methods. So of course, your traditional harp trapping. We also used canopy. Obviously, there's not really a canopy in the arid zone, but we use these at height mist nets. These are particularly effective at catching the Aussie mops. And I did incorporate acoustic bat lures as well, which I did my master's. A lot of that was on acoustic bat lures. So this just brings the Aussie a bit lower so you can catch them in these nets. And so from this capture data, I'm also going to be looking at, so I've collected all the bat scats, and I'm going to look at diet and look at how this differs inside and outside the fence to see if there's any sort of resource availability issues. I'll also incorporate some radio tracking work, and I was pretty pleased with this. So I mentioned that Aussie mops, Peter's eye, is a beautiful, cute little bat. They are until you try and stick something on them. Then they go crazy and they just preen it off. They won't fly off until they preen it off. So I developed this collar. So the bats I trialed this collar. Unfortunately, it was hampered a little bit by rain. Again, lots of things I did was hampered by rain. But this worked really well. So these are actually, ironically, cat gut sutures. So they naturally break down in the environment after about a week. And they tested them in temporary captivity. So I kept them for a full day and made sure they were happy with the collars and the tightness and everything. And they did relax quite quickly after they were put on. And we got some really great results because we were really struggling to get results from these bats. So we now, and this isn't fully updated, but we've located all of these roost sites. Most of the trapping was inside the reserve, so it's a bit of a skewed, I guess, but we can see that bats generally seem to have quite high roost fidelity, and they do then roost outside a bit, but generally there are these 2 hotspots inside the fence. And we think this is why at Wild Deserts we see more activity inside, because we've sort of captured these 2 fantastic areas just with inside the fence. They very rarely will go outside. And this is a place called Beefwood Boulevard. So this is the top site, that top cluster. So these are these big beefwood trees, which are quite rare in the area. They're not that prolific. So we think that these are really important for these bats, and they are known to be predated on, or not predated on, but eaten by rabbits a lot. So there is, in 100 years or so, maybe we'll see a lot more of these in this area. So we tracked the Aussie mops to these roosts. And we tracked Nyctophilus to the mulga trees. They will just roost under exfoliating bark. But what we then did is found the roosts. We used endoscopes to see how many bats were roosting in the trees. We took all of the measurements of the aspect. We've also put eye buttons in these roosts to see if there's sort of any thermal buffering or the conditions inside the roost for both species. So yeah, my final chapter will be incorporating all of this. I also have 2 other very large scale grids, which I put inside the fence. And then I have other vegetation data. So they'll be looking at really how they're utilizing the space inside the reserve. So more to come, hopefully next year, you'll see some of that published somewhere. And I'm just going to finish up with a video for you. So I mentioned all the rainfall that I've experienced since my PhD started. And if we go back to this slide, you'll see, it's not in this one, but there's a very large lake. So this is a big swamp just outside. About another 3 kilometers to the east, there's a very large lake, which is generally always dry, only sort of fills once every anything from 10 to 40 years. Some people have lived there for 30 years and never seen water in the lake. I've seen water in that lake twice in my 4-year PhD. So this was 100 millimeters of rain, so half the yearly average that we received in about 36 hours. And this filled the lake. I'm not sure if you've got sound, but you don't really need it. And of course, I'll just finish by thanking my many sponsors that have helped me out a huge amount throughout my PhD, both financially and with other support. So yeah, thank you very much, and happy to take any questions that you may have.
Thanks, Oli. That was super interesting and great to see all the different aspects of your PhD and how they are developing. We don't have any questions in the chat, but I can see a couple of hands up. So I'm just gonna ask people to unmute and ask their questions. So Felix, I think your hand went up first. So if you want to unmute and ask your question, please.
Yeah, thank you very much. Thanks for that talk, Ollie. It was very interesting. I love the diversity with all your different chapters. Seems really exciting to do that research. I also joined a little bit late, so sorry if I missed this. But when you were doing your halo effect chapter, were you doing habitat surveys around each detector to look at like vegetation characteristics, or was that not an aspect
Yeah.
for that element?
No, we were. So obviously with time and the amount of sites, it was a fairly basic. So we used the clutter index, which was a measure of habitat complexity. We didn't do any sort of fine scale vegetation assessments. It was really just this idea of habitat complexity and that sort of clutter. As you can imagine, the areas are relatively homogeneous. We also did standardize every site because we didn't want to have that sort of bias by the sites. I mean, still the site variation explain the most variance in the model, of course. So there's something else going on there, but yeah, so kind of, but not in depth. It would have been nice to incorporate that a bit more, but yeah, it's just sort of a limiting factor a little bit.
Okay, the clocker index, you say then?
Clutter, sorry.
Oh, sorry, the clutter. Sorry, the clutter index.
Yeah,
Okay.
so you take
Was that just with a stick, like a augmented
well,
stick, or?
you use a measuring stick, but you essentially look at different height classes.
Yeah,
So
yeah.
bare ground, not 25 centimeters, 25 to 50.
Okay. Yeah.
Up to 2 meters or above 2 meters.
Yeah.
And then you take a percentage cover
Yeah. Yeah.
of that height, and then you convert that into a single number as your index of clutter.
Yeah.
And it's all that, it was significant for the clutter bat, which makes sense. And yeah, I think that's a big part of it. And there's these other factors, you do have the lake, which then we had this rain come down. So the open space bat and the edge space forager, I think they actually move away from the reserve. Because, the conditions, I mean, I first drove out to the Arid zone and I was like, wow, this isn't very like a desert, it's more like a prairie. So I think the conditions are so good, it may have masked some of our effects. And when you look at the more long-term data where we do actually have those conditions in drought and just after drought, we do see more of a difference between inside and outside. So it'd be great to do the halo again. Selfishly, my last year, it was predicted to be a drought year, and I kind of wanted it to be a drought so that I did have that data. But it's a good thing that there was lots of rainfall again in the arid zone. But yeah, I think that was may have masked some of the benefits of the reserve.
Cool. Thanks.
Thanks.
Thank you, Felix, for your question. Eleanor, you have your hand up as well.
Oops, I do. Thanks very much. Yeah, lovely study, Ollie. That's fun stuff. Are you going to be doing insect surveys apart from what the bats are eating? Because in my experience, bats, I mean, it's all nice to say they fly and clutter, etc., but they choose the food they want to eat first. So, for example, if you look at our monogam stuff in Eswatini, the bats were deliberately leaving the nice proper bush areas and going into the sugarcane areas because there's lots and lots of insects to eat there. So you go where the food is.
Yeah, so I did do some sampling over about a week inside and outside the fence each night. So using light traps. And I do have those insects in my freezer currently, actually. But to be honest, I found that the nightly variation was so huge. Like some nights there would just be absolutely, I'd get there and there would just be this plague of insects around the light traps. And other nights it would be absolutely dead. I did want to try and use more of a biomass rather than try and identify everything. But yeah, I just feel like there was such nightly variation and site variation. I have still got the insects. It's something I could still do, but I kind of wanted to look at the diet from the bat scats first. Yeah, and it also kind of quite quickly maybe got put in the too hard basket, to be honest. I've liked to have done more, but it's quite a lot of work, and particularly then to go through and identify each. I mean, even if you just go down to sort of order. So I think biomass would be good, or just to filter into different size classes. I guess the short answer is kind of, but I'm not sure if I'm going to incorporate it at this stage.
There's always another question that comes up, isn't there?
Yeah,
because
exactly.
My next question was, does that biomass in any way relate to their activity? Because again, in my experience, if you've got something like termite, a late swarm, your bats are out for 10 minutes and they've stuffed themselves in 10 minutes and then they go back.
Yeah. Yeah, and I think it was also my lack of knowledge around invertebrate sampling and maybe how to do it properly and to incorporate that with the bioacoustics or I mean the trapping data is not really that great. Yeah, I think in all honesty, I think it just got put in the too hard basket a little bit. But it is something that I wanted to look at more, but perhaps missed the boat a little bit with that one.
If you've got bioacoustic data, you've got feeding buzzes.
Yeah,
you can actually tell whether they're feeding.
Yeah, so I did try and separate out some of the feeding buzzes. One of the issues out here is that there's very low call rates as it is. Another reason why we just use these 3 focal species. We're getting very low calls compared to what you'd get in any sort of temperate zone or tropical area. So I did start pulling out the feeding buzzes, but the numbers were so low that it wouldn't really have been anything. It wasn't really usable data.
Okay,
but great
super.
question.
Thanks.
Yeah, I probably should have done more with that.
There's always another question, and that's half the fun of working with bats.
Yeah.
Great stuff. And that actually relates to a question that I had about the sort of spatio-temporal patterns that you detected in your first chapter. I'm guessing you're going to explore that more with the radio tracking data that you have as well. So you think maybe the bats may be sort of using the reserves probably more for roosting, and then not really caring that much about the reserves or even going outside the reserves when they're looking for food. So I just wonder what you expect to see with those radio tracking data.
Yeah, we had quite a few issues with the radio tracking data. I think they're traveling a lot further than we expected. A lot of the bats, when we were tracking at night, they would come out of their roosts, and we had a sort of team of 6 different base stations with yagis. So they'd come out, they'd forage around the reserve for maybe an hour and a half, and then they would literally disappear into the nether. And we would drive kilometers in each direction and just completely lost them. So I think they're traveling, the home range of these bats is meant to be about 10 kilometers. But yeah, I think they're traveling a lot further than that. Certainly with the Nyctophilus from those gams, you can see that they are just heading out. That swamp that I highlighted, I think is a really great, and obviously it's not generally wet, but I think that's a great foraging area. Certainly when you drive around the swamp, the insects are just all over the place. So I think for those guys, that's why we saw that effect, because they're just not traveling that far. There's smaller insects. But I think for the Ozymops and the Calanolobus, they're really traveling out towards the lake where there's a lot more dead large trees, and then just coming back to the reserve. And particularly even in those very wet years when there's water in the lake, I think they pretty much just all head that way. And maybe the reserve then is a bit more of a refuge when it starts to dry out. And maybe that's related to the reduced herbivory. I'm not entirely sure. It's hard to sort of pinpoint exactly what it is.
Yeah, very interesting. Really looking forward to seeing those results more developed. Anyone else has any other questions for Oli? Some comments in the chat saying very nice presentation. Actually, I had another question about one of the plots that you showed. I think it was the one related to your second chapter where you were comparing activity levels inside the reserve and outside the reserve. And there were higher levels inside the reserve, but only the first couple of years, and then they seem to
seem to even out.
Yeah, so
Yeah.
I just wonder why you think that was.
I think during the latter years there was, it was sort of a bit of rain came down, and we see that initial increase. I think the inside the reserve flourished very quickly. That is slightly lower in the water table as well, some of those areas. But then I think in latter years we had much higher rainfall, and that's when the lake started to fill. And again, similar to what I was saying before, then I think it actually draws bats away from the fenced area. Across the whole wider landscape were much nicer. So maybe then the reserves stopped having any kind of difference in conditions compared to outside.
Okay.
Yeah,
that's really interesting as well. Just depending on.
Yeah, it's really hard to pull apart how much of it is the fence and how much of it is just the habitat itself and where it's located. And obviously with such a mobile species, it's quite hard to really pull that apart.
Yeah, but presumably those differences in habitat will be more obvious as time goes by.
Yeah.
And the effects of the reserve being there become more
Yeah, for sure. And it's been cool to start getting into this remote sensing data with the vegetation and not want to explore that a lot more. And one day, it'd be great to put GPS trackers on these bats and really see what they're doing, maybe in a few years.
Brilliant. Okay. Well, if there are no more questions for Oli, I think we just have to thank you again for your presentation.
Thank you very much.
And for answering our questions. And thanks everyone who joined us today. And just to remind you that we'll have more webinars in the next few months. So we hope to see you again. So yeah, thank you everyone and enjoy the rest of your afternoons.